So let me now first of all review the program that we have a short introductory talk on what our SciLifeLab COAVE COVID-19 research program is all about. And then we have 3 guest speakers, and at the end of the day, a panel discussion on the future of drug discovery in this space. Let me just start from the SciLifeLab angle, mostly for our international guests here, as to what is SciLifeLab. So we are a national life science infrastructure and a collaborative research center that is organized by the 4 host universities, KTH, Karolinska, Stockholm University, and Uppsala. And this was established about 10 years ago, has had a national status, and we have sites all around Sweden and do work across Sweden these days as a national infrastructure. And we are now having our 10-year anniversary, made a new strategy incorporating new things. So today we are about infrastructure, research, and data for the life science. Well, this year, obviously, COVID hit, and we were obviously considering what should SciLifeLab do as a national player in the field of COVID. So first of all, we prioritize our infrastructure for users that had COVID-19 questions. And then we were able to work with the Knut and Alice Wallenberg Foundation to raise funding for a research program on COVID-19 and set up a virus testing facility, high throughput and high content serology testing, environmental virus profiling, a national data portal. And now as a result of all this action, lots of research breakthroughs, biobanking, and these types of national meetings that we are now witnessing today. And all the time, this has been characterized by rapid action, integrating people, integrating and coordinating capabilities, and we want to continue this. Excuse me. So this is how the program looked like in the beginning in April, 67 funded projects that we classified in different areas. And today's meeting is really co-sponsored by these 2 areas, drug discovery and repurposing of drugs, and the host cell systems biology and targets. And altogether, this is really a unique program in our mind, that we had a very rapid turnaround time to select this, the participant in this program. And instead of just funding individual PIs, since we are not really a funding agency typically, we asked scientists to collaborate with each other and coordinate each research area, provide funding to biobanks, provide funding to SciLifeLab infrastructure to boost the capabilities, and created a national data portal for sharing COVID-19 results. And now all universities in Sweden participate in this program. And as part of this program, there's been also the setup of high throughput viral testing. So some 500,000 people have been tested, some 50,000 people have received a diagnosis through this program. And as you see, we continue to have a high prevalence of COVID-19 positivity as of this week, and it doesn't go down. So we probably will continue to see high numbers of cases in Sweden. We've set up biobanks across Sweden for collecting samples from patients. And we have created this COVID-19 data portal that integrates all research, both our research and those funded by any other funders. And this was done together with Vetenskapsrådet, the National Research Council that integrates nicely all of the data. So I think this has shown how we've kind of organized the research in Sweden in this setting. So just now at the end, since we have people who have participated in this program, and they may be wondering as to what happens next. But we have been very, very happy and lucky to get another set of funding from the Knut and Alice Wallenberg Foundation to continue the COVID-19 research in the next 2 years. We've got a second separate allocation of funding for studies of vaccination effects. We are hopeful that the new budget for SciLifeLab includes funding for laboratory preparedness for pandemics, which would amount to 100 million krona. And then, as many of you heard, we recently got a 3 billion krona donation from the KAB for the data-driven life science program. And as one of the topics there is epidemiology and infection biology. So we want to now make use of everything that we can in terms of supporting database and data-driven ways of combating the COVID. So the plan is actually this week to announce a new call for the continuation of this funding. So altogether, this is about 200 million of dedicated support for research and preparedness for pandemics. And this will be in the form of 5 different types of calls, continuation of the existing research with new constellations and collaborations across the research areas, new single PI grants, then a specific program looking at the effects of vaccinations as they are being launched next year. Then ideas on how pandemic preparedness at SciLifeLab should be handled in the future, and ideas on how data-driven research on COVID-19 related topics could happen. Idea is that we sort of have a proposal deadline in January, and we'll go through a fairly quick review. I don't want to put a date now, but hope to get decisions in February, and also coordinate these activities with VR, particularly regarding the vaccine program. So there's a lot of happening in this space, and I hope many of you will be interested in applying and participating in a stage 2 COVID-19 research program from SciLifeLab. And that was all that I had to say. So I will now thank you for your attention, and I will pass it over to Anna-Lena to give her starting words. Once again, thank you, and look forward to your an interesting seminar and discussions at the end. Thanks.
So thank you, Olli. And again, welcome to this half-day meeting on accelerating the process for basic drug discovery to clinical trials for COVID-19. My name is Anna-Lena Gustafsson, and I'm the Director of Chemical Biology in Genome Engineering Platform here at SciLifeLab, and also one of the scientific leads for one of the research areas. So I will take the opportunity to introduce and give a short overview of the projects in the area of drug discovery, drug repurposing, and mapping host cell target interactions within the COVID-19 efforts at SciLifeLab. So first, a general introduction how SciLifeLab tries to contribute to drug discovery. And drug discovery is the process by which prototype drugs are discovered or designed. And we try to build awareness of the need for academic science in this process by supporting pre-competitive research, build network, and facilitate communication. We also work hard to secure quality of data, as this is important to generate reproducible results. We also try to promote the use of frontline technologies that are available at SciLifeLab platforms. And last but not least, SciLifeLab has an infrastructure that is fully dedicated to support drug discovery projects. So at SciLifeLab, we have a number of ongoing projects that you heard Olli mention in these areas. And for drug discovery, repurposing, and host cell interactions, we have tried to cluster these into 3 areas. Targeted-based drug discovery. Here we have 3 project examples targeting MPRO. And this has resulted in identification of several new chemical series that are active in vitro and also in various cell models. We have 3 projects under the area of profiling for drug repurposing. And these projects use both targeted phenotypic imaging assays and also untargeted approaches, such as combining cell morphology like cell painting with gene expression analysis. 12 projects addresses host cell interactions, which could be based on phenotypic high content assays or proteomic studies or even CRISPR screens. And these projects are of course important as they are the basis for the possibility to identify new targets. And the areas of interest are listed to the right in the slide. And you can find more information on the webpage. So based on all these ongoing projects within SciLifeLab, addressing drug discovery, phenotypic screening and profiling, and target identification, we are setting up a chemocentric database to take care of all the data that is generated internally. That will also be communicated to the scientific community through this data portal. And based on the ongoing projects, we also tried to build a structure based on the SciLifeLab infrastructures and platform that will be a value not only to the ongoing project, but also for the possible future coming pandemics as well. And on the final slide, we try to illustrate what is ongoing in terms of contacts and networking activities, as we see this, of course, as an international collaborative network for drug discovery. And we are very much interested to hear what the presenters say here today, and we will also take the opportunity to hear what you think would be the next step for the projects and how SciLifeLab can make an impact. And we look forward to the presentations and the panel discussions, of course. So by that, I would like to hand over to my co-chair, Johan Lennerstrand, who will introduce the first speaker.
Hello. So I'm very happy and honored to present Daniel Kuritskas, whom I've known since 20 years back from HIV drug-resistant workshop meetings. Daniel is a professor in medicine at Harvard Medical School. He's the chief of division of infectious disease at the Brigham and Women's Hospital in Boston. He has published extensively on antiviral therapy and drug resistance and HIV. He has chaired several multi-center studies on HIV therapy. He serves as a member of the NIH Office and AIDS Research Advisory Council, and as a member of the United States Department of Health and Human Service Panel on guidelines for antiviral therapy. So his research is focused on HIV therapeutics, antiviral drug resistance, and HIV eradication. However, this year, Daniel, like many others, have switched viruses. And I've been listening to these virologic education webinars during the year, and it was very impressive talks by Daniel on COVID-19 clinical trials. So I would like to hear something similar today. Daniel, the word is yours. Thanks.
Well, thank you very much, Johan, for that very kind introduction. It's a pleasure to be here and to speak with everybody this morning. Let me share my screen and get started here. And just bring this to slideshow. Very good. So today I'm going to talk about clinical trials on antivirals for COVID-19, what has been done and what is expected to come. These are my disclosures. I'm going to cover principally four topics and then touch very briefly on the fourth. So I'll talk mostly about what we know about RNA-dependent RNA polymerase inhibitors as COVID-19 therapeutics, principally remdesivir, and then say some words about the use of convalescent plasma, which is really a generic approach to the much more refined and specific approach of the use of monoclonal antibodies. And then I'll wrap up with mentioning some drugs that we know don't work. So this slide, which is really courtesy of Thomas Szilard from Gilead, that was presented at a recent NIH workshop on antiviral therapeutics for SARS-CoV-2, lists the RNA-dependent RNA polymerase inhibitors that are currently in human clinical trials. Remdesivir, which is now approved for treatment of COVID-19 in the United States, has of course completed phase 3 trials, whereas there are 3 other agents that are in earlier stages. Molnupiravir, which is also known as EIDD 2801, a Merck compound, is now in phase 2. Favipiravir has undergone phase 3 trials in Japan, but these are very small trials that really would not qualify as phase 3 in some other countries. And although they plan to file in Japan for approval, it's doubtful that the data that they've generated to date would satisfy the US FDA. And then AT527, which is being developed by ATIA, is a drug that initially was shown to have activity against hepatitis C virus in phase 1-2 trials, and is currently in a phase 2 trial. And by way of disclosure, I helped to design that trial, and it is still in phase 2, so there's not much that can be said about the ongoing study. And lastly, galidesivir is another RDRP inhibitor that is just entering into early stage human trials. So really, I'll focus much of the time in presenting the data that have been collected on remdesivir, which as more data accumulate are less and less clear about exactly just how effective remdesivir is. This is a compound that has in vitro activity against a wide range of viruses. It's been studied for Ebola, SARS, MERS, and now, of course, SARS-CoV-2. And interestingly, in clinical trials published in the New England Journal of Medicine a year or so ago, remdesivir was found to be inferior to broadly neutralizing antibodies against Ebola. But remdesivir does have activity against MERS and murine and non-human primate models, and was shown to be effective against SARS-CoV-2 in the non-human primate model, and in vitro has inhibits SARS-CoV-2 with an EC50 of a little bit under 1 micromolar. The first data to be reported on remdesivir in patients with severe COVID-19 came from a randomized phase 2 trial that was conducted in Wuhan in China, which looked at time to clinical improvement by day 28. This trial was initiated just as the epidemic in Wuhan was coming under control. So only a little more than half of the planned 450 participants were actually enrolled, dramatically reducing the power of the study to find any significant differences. And as you can see, there really was no difference in time to improvement between the remdesivir arm shown in red and the control arm shown in blue. Likewise, there was no appreciable effect on the decay in virus recovery from either the upper respiratory tract shown in panel A here, or in the lower respiratory tract shown in panel B. Oops. Here we go. But then there were data that came from an open label experience, compassionate use experience in patients with severe COVID-19 in the United States. These data, of course, have no comparison except for contemporaneous experience in patients not receiving COVID-19. What you see in panel A overall is the rate of clinical improvement for all participants who received the drug, and the median time to improvement was just under 3 weeks. More notably, for those who required oxygen support at the time they first initiated remdesivir, particularly those requiring invasive support, meaning mechanical ventilation, the overall median improvement was about, again, just under 3 weeks, and with 50% of participants showing improvement at a time when the survival for intubated patients was at around 30%. And so by comparison, these patients appeared to be doing better. The benefits were clearly much more pronounced in younger people than in older people. But again, this is a non-comparative study. The first randomized controlled data came from the ACT-1 study, a study sponsored by the United States National Institutes of Health. This was a placebo-controlled double-blind trial in hospitalized adults with COVID-19 pneumonia. They were randomized evenly between remdesivir or placebo, and the primary endpoint was the time to recovery within the first 28 days. And this analysis was conducted after there had been just over 600 recoveries. And you can see that overall, there was a benefit that was statistically significant, albeit somewhat modest, about a 3 to 4 day faster improvement in the participants who received remdesivir compared to placebo. This benefit was most notable in those already receiving oxygen, less clear-cut in patients who were well enough not to need supplemental oxygen. And there was really no obvious benefit in those who were receiving either high flow oxygen or required mechanical ventilatory support. In a subgroup analysis, here again, you see overall, there was about a 30% improvement in the recovery rate ratio that was statistically significant. The effect was more pronounced in younger than in older patients. There was also somewhat greater benefit in Caucasians than in Blacks and Asians. And then looking at the baseline ordinal score was again most notable in those who were receiving oxygen, no obvious benefit in those receiving mechanical ventilation, and less clear-cut benefit in those who were not yet receiving supplemental oxygen. So there were then 2 studies conducted by Gilead, known as the SIMPLE trial. These were the one done in severe disease and one done in moderate disease. The severe study was a non-controlled study. This study simply compared 2 different durations of remdesivir administration, either 5 days or 10 days, with the primary endpoint being the clinical status at day 14, using this same ordinal scale that categorized people by just how sick they were and how much supportive care was required. Patients or participants had to be SARS-CoV-2 positive by PCR, have evidence of pulmonary infiltrates, that they needed to be hypoxic, and could not be receiving mechanical ventilation or extracorporeal membrane oxygenation, ECMO, at the time of enrollment. The study, which has been published in the New England Journal of Medicine, essentially found no difference between the 5 and 10-day groups in terms of clinical status at day 7, time to clinical improvement, or time to recovery, which meant really time to discharge or preparation for discharge from the hospital. However, a contemporaneous cohort was also studied at the same time. This was done in centers that were not conducting the trial, and so the participants there had no access to remdesivir, but were otherwise reasonably well matched to the people who were in fact in the trial at other centers. And in that analysis, it was found that the time to recovery, or the proportion recovering who in the remdesivir cohort was significantly greater than in the non-remdesivir cohort, the 75% versus 60%. And also the mortality at day 14 was lower, about 7.5% versus 12.5%, and both of these differences were statistically significant, but again, not a controlled clinical trial. So the SIMPLE trial in moderate disease was a controlled trial. This was a 3-arm study that once again compared 5 days versus 10 days of remdesivir, but also with a standard of care arm. Once again, the participants had to be SARS-CoV-2 positive by PCR, but here they could not be hypoxic. They needed to have an oxygen saturation of 94% or greater, requiring no more than 2 liters to maintain that level of oxygenation. And initially, 600 participants were enrolled, although subsequently the study was expanded as an open label study once the primary results were available. The primary endpoint was clinical status at day 11, and the 5-day arm showed a greater clinical improvement compared to standard of care with an odds ratio of about 1.6, which was statistically significant. Now, if you look at the different outcomes between the 3 arms in this study and the different days, so here was the primary endpoint at day 11, and this is just graphically illustrating this ordinal scale where the lightest color here is being discharged and the darker colors are either still being hospitalized or the darkest color actually having mortality. And at day 11, the distribution of endpoints is significantly superior for the 5-day arm compared to standard of care, but curiously not for the 10-day arm. At day 14, both arms show a benefit compared to the standard of care. At day 28, things have switched, and now the 10-day arm, but not the 5-day arm, are statistically significant. So evidence that remdesivir is probably beneficial, but some confusion about which arm is actually statistically significant compared to the standard of care. Well, then the waters get a little bit more muddy when the Solidarity study was conducted or reported out just several weeks ago. This was a much broader study. It had actually multiple different arms, and this analysis that I'm showing here is limited to the remdesivir arms. The participants were eligible if they were hospitalized adults with COVID-19 who had not yet received the study drug. The primary endpoint was in-hospital mortality, not improvement in clinical status or change in this ordinal scale. So a very different endpoint than in the other studies we've discussed. But a secondary endpoint was initiation of mechanical ventilation and duration of hospitalization. You can see that about 2/3 of the participants needed oxygen at entry, about 10% were already ventilated, and a quarter were healthy enough, despite being hospitalized, that they didn't require supplemental oxygen. There were just under 5,000 participants who were in the intention to treat analysis for the remdesivir component of this trial. And what the study showed was no difference. You can see here that as compared to control for mortality, there's absolutely no benefit for remdesivir when the data were analyzed by different subgroups, either by age, whether participants were ventilated or not ventilated at the time of entry, and then again overall, no difference for remdesivir. The authors of this study then did a meta-analysis that included data from other randomized trials of remdesivir and find that overall little evidence for benefit of remdesivir across a variety of categories, whether here just for the Solidarity study looking at oxygen requirement at entry, here for the ACT study, again looking by oxygen requirement, where the only benefit as seen in the initial study was with the low flow O2, and then overall a trend towards some benefit, particularly in lower risk groups not yet requiring ventilation, but just missing statistical significance. So where does that leave us for remdesivir? Well, for patients with mild or moderate COVID-19, the US Department of Health and Human Services panel, the counterpart to the HIV guidelines panel that Johan mentioned before, concluded that there are insufficient data to recommend either for or against the use of remdesivir in patients with mild or moderate COVID-19. Patients who require low flow supplemental oxygen should get remdesivir for 5 days or until hospital discharge, whichever comes first. No need to keep a patient in the hospital to complete a course of remdesivir. And for those who require higher levels of oxygen, either by delivery through a high flow device or mechanical ventilation, the panel concluded that there's uncertainty regarding whether or not remdesivir provides a benefit. The World Health Organization, by contrast, leaning heavily on the Solidarity data, really conclude that there's no benefit or only very weak evidence of benefit of remdesivir and do not recommend remdesivir for use. Well, let's turn now to talk about antibody therapy. And first, we'll talk about more generic uses of antibodies in the form of convalescent plasma. A convalescent plasma from survivors of COVID-19 infection may contain high titers of neutralizing antibody, and convalescent plasma has shown some benefit in the treatment of avian influenza, H1N1 influenza, and MERS. And pilot studies and uncontrolled trials suggest a possible benefit of convalescent plasma, which I'll now call CCP for convenience. And the largest data set we have come from these preprinted data from Joyner and colleagues describing the experience of the Mayo Clinic group with this expanded access program. This was a program that was undertaken really to facilitate access to convalescent plasma throughout the United States without each center having to design and submit to the FDA their own expanded access plan. And to everybody's great surprise, this enrolled a huge number of participants. Over 35,000 are reported here. More than half of them were in the ICU and more than a quarter were intubated. There are no control data here because everybody received convalescent plasma. So the comparisons really are between earlier versus later receipt of therapy, receipt of high versus low titer antibody, and then looking at transfusion, the 30-day mortality and 7-day mortality. So in the 7-day mortality, what you see is that those who had low titer antibody had higher mortality than those who received high titer antibody. The same trend is seen for the 30-day adjusted mortality. And then if we look more in more detail at those who received treatment within 3 days versus 4 days, in those treated earliest who got high titer antibody did have a significant reduction in mortality compared to those who received low titer, and there's no such difference in the 4-day group. An additional analysis was conducted by the FDA looking at these data and neutralizing antibody titer assays performed by the Broad Institute. They found no difference in the 7-day mortality overall, but did see a significant reduction of about 20% among non-intubated patients who received high titer antibody compared to those who received low titer antibody. There was no benefit in the more severely ill participants who were already intubated at the time that they received CCP. And in those who were not intubated and under age 80, receipt of high titer convalescent plasma within 72 hours of admission was associated with a significant reduction in 7-day mortality, nearly a 50% reduction, which was highly statistically significant. But again, no control group. Now, just recently, last Thursday, in fact, the PLASMAR study conducted in Argentina was a placebo-controlled randomized trial of CCP for COVID-19. This study enrolled 334 participants, randomized 2-to-1 to receive CCP versus placebo. The plasma had to be titered and had to have a titer of at least 1 to 400 for COVID-19 specific IgG. The median titer was actually, 1 to 3,200. That's a typo on my slide. It was tenfold lower than shown here. The median time to symptom onset was 8 days, so a little longer than the period that was found to have benefit in the Mayo Clinic experience, and maybe explaining the negative results that I'll show you in a moment. 90% were receiving supplemental oxygen, and almost all had received corticosteroids. But the primary endpoint was the distribution of outcomes at day 30. And here you see the primary endpoint. There's no difference. Whether looking at 30 days, 14 days, or very early on after the intervention, these distributions using the same ordinal scale that I showed before had no significant difference. So uncontrolled trials of CCP suggest convalescent plasma may provide benefit, but the benefit appears related to the very early administration of high titer convalescent plasma. There's little or no benefit in critically ill patients. No benefits observed in the PLASMAR study, although there are other randomized trials that are still underway. As a consequence of these data, the IDSA guideline panel has recommended that COVID-19 convalescent plasma should be used only in the context of a clinical trial. And the DHHS treatment guidelines state that there are insufficient data to recommend for or against its use, and that the CCP should not be considered standard of care at this time. Well, that brings us then to monoclonal neutralizing antibodies. We know that these antibodies can neutralize SARS-CoV-2. They all target the S or spike protein. They can be genetically engineered to either combine multiple specificities or by modifying the Fc domain to extend half-life. And they have potential utility for treatment or prevention of COVID-19. And what we've seen to date is really a smattering of preliminary data, which have led really, I think, inappropriately to issuance of expanded use authorizations in the United States, and the very likely possibility that we will never see a real compelling data from a phase 3 trial. The first of these is the BLAZE-1 study for a Lilly antibody that is now known as bamlanivimab. This was a placebo-controlled phase 2 trial that had as its primary endpoint the change in virus load at day 11. And that endpoint was met for the middle dose group. There was also a reduction in visits to the emergency room or hospitalizations, that was about a threefold reduction. There were no study-related serious adverse events, but there were some resistance mutations that emerged, or at least putative resistance mutations, but they seemed to emerge whether the participants received antibody or received placebo. And at the time, there were still ongoing trials. These data show the three arms from the monotherapy study with bamlanivimab. I never remember, it's easier to say CoV555. And then this is the combination study that is still underway. Note, we have no data for this second antibody by itself. And here you can see for the symptom score that there were significant reductions in symptom score for people who received these antibodies, but this is all in phase 2. Now, Regeneron has a cocktail. This is the cocktail, of course, that Donald Trump received when he was hospitalized with COVID-19. And there's an ongoing seamless phase 1 through 3 trial of this cocktail, and the constituents are now called casirivimab and imdevimab. This study, at the time of its reporting, was really just phase 2, had just under 300 outpatients. I should mention, for both the Lilly and the Regeneron study, these are outpatients. The studies in more seriously ill inpatients have been stopped for lack of benefit, and there are still ongoing studies in less seriously ill hospitalized patients. But in this outpatient study, there was a significantly greater but modest acceleration in decline of viremia, and there was avoidance of hospital emergency room visits or hospitalizations. That was about 50% overall, and maybe a 30% reduction in people who were high risk. But note, these are very small numbers, so really not compelling evidence. So to summarize, for the monoclonal antibodies, there are numerous antibodies that have been produced. Several of these are now in clinical trials. They have shown benefits in animal models, and there are preliminary data for the Lilly antibody and the Regeneron cocktail that appear encouraging, and no safety concerns to date. And now there are emergency use authorizations for both the Lilly and the Regeneron antibodies, but we have no phase 3 data, and there's a serious concern that we will never have phase 3 data, and we'll never really know if these antibodies have benefit or not. I'm going to end by just summarizing very briefly 2 drugs that don't work. We know that hydroxychloroquine has no benefit. Multiple studies have shown that. And there are now several studies that also show that lopinavir/ritonavir has no benefit. And there are in vitro data suggesting that other protease inhibitors, notably darunavir, has no activity. I haven't touched on it for the sake of time, but protease inhibitors, inhibitors of human protease inhibitors like the TMPRSS2, which is necessary to process the spike protein for entry, are underway. Camostat is now being studied in several trials, and more are planned. I'm a little skeptical about this approach, only because TMPRSS2 is not the only protease that can process spike. Cathepsin can also do so, and at least in vitro, if you inhibit one protease without the other, you only have a modest reduction in infectivity. And so it will be interesting to see what the Camostat studies show, but I think that other approaches are probably going to be more fruitful. So I will stop there, and thank you for your attention, and I'm happy to take questions. And I'll stop sharing my screen.
Thank you, Dan. I have 1 or 2 questions, and then we'll see if anyone can send in on chat also. I haven't seen anything here. So I'll start with, well, timing is everything. So this is the acute virus. If the patients would have taken remdesivir earlier on, I mean, it's intravenous, they come to the hospital. The trials don't really see what would happen if they had taken a quick assay at home and taken a pill instead, and lessons to be learned from the flu virus with Tamiflu. what's your opinion on this?
I think you're absolutely right, Johan. I mean, everything we know about respiratory viruses, and certainly the experience with influenza is very informative here, teaches us that the greatest benefit is seen when people start treatment immediately, really within the first 24 to 36 hours of the onset of illness. Of course, with influenza, we have the benefit that, at least for most patients, the onset is pretty abrupt. And so you know when you got sick with influenza, and you can get your oseltamivir or your baloxavir right away. Onset for COVID is a little bit more insidious, and there are now ongoing trials of remdesivir, one in outpatients for an abbreviated course, just a 3-day course. The fact that the drug needs to be given intravenously is somewhat of a disadvantage here. There are plans with the AT527 compound that if phase 2 is positive in terms of its virologic effects and safety in hospitalized patients, to move to an outpatient trial. And there is at least one Gates-funded study that's planned with remdesivir for a single dose for outpatients. And then the Merck compound that I mentioned is also being studied in outpatients. But I think the earlier we intervene, the likelier we are to see benefit.
So you see a future for oral pills in combination, like HIV and hepatitis C?
That's an interesting question. I think it's going to be more and more challenging to do these combination studies. Well, first of all, I think combinations would be important only if we see that resistance becomes a major problem. We were able to treat influenza really with single agents without much of a problem. And now that a vaccine is clearly on, or several vaccines are clearly on the horizon, I think there is the hope that we will see, at least by the end of spring, significant reductions in the rates of COVID-19 that will make conducting these trials more challenging. That said, I think there is still going to be an ongoing need for therapeutics, because we know not everybody is going to accept the vaccine, and we have to anticipate that despite the very encouraging preliminary trials, we can't be certain that vaccine will be effective in all populations, especially in very elderly or immunocompromised patients, where we may continue to see infections and continue to need therapeutics.
Yeah, we would see maybe there will be resistance, even though this virus doesn't mutate as much as the other HIV and hepatitis C. It has an error-prone, it has an editing ability, and it's an acute virus. But what do you think? Could there be resistance? We have seen some Tamiflu resistance, and what? You never know.
Yeah, I certainly think that we have to be concerned that resistance may emerge, that there are ongoing efforts to collect samples from people, especially those who have persistent viremia after treatment with remdesivir and other drugs, to see if there are changes in the polymerase that may be associated with a shift in susceptibility. Doug Richman, whom you mentioned when we were chatting before, has said that the definition of an antiviral drug is one that selects for resistance. With widespread use, I think we have to anticipate that there could be resistance. I think it would be helpful to have drugs that target different parts of the virus life cycle, and drugs that have non-overlapping susceptibility patterns to anticipate that.
Yeah, then there would be a need for combination treatment. And in the third world, well, maybe the vaccines are not accept, if it's going to be a cold chain, etc. For some vaccines, maybe there would be generic drugs available in the future for these developing countries.
Well, I hope very much that the model that has been set with HIV, where innovator companies that have patents for the sale of their drugs in higher income countries are willing to license for local manufacture and distribution at access pricing these drugs in lower and middle income countries. And we've seen really dramatic benefits from that approach with the widespread use and availability of antiretroviral drugs. And I think that has now established, I think, sort of the ethical framework in which new drug distribution should be made available. We've seen that to some extent for the hepatitis C drugs as well. And so hopefully we'll see that for drugs for COVID-19.
Yeah, thanks. I have no more questions. I think Ollie has a question here. Where?
I can also.
Yeah, good. Take over.
Thanks for the talk. That was fantastic. So it was painting a rather pessimistic picture on the antivirals. So what made me to wonder as to how much has the dexamethasone survival benefit that is quite remarkable. How much has that impacted on the antiviral clinical studies? And how do you view the role of dexamethasone before or after or during the antivirals?
Yeah, that's a great question. I didn't cover the anti-inflammatories because it's a whole other talk. But yes, the only drug which has clearly shown a survival advantage is dexamethasone and other corticosteroids similarly. Those benefits, though, are really in people with moderately severe or worse disease. So at least in that moderate category, those are patients who are getting both steroids and remdesivir. So people who have an oxygen requirement and are not yet severely oxygen dependent would fall into this zone where they are eligible for remdesivir and eligible for corticosteroids. And we are using both of them now routinely. I don't think the availability of steroids has made it that much more challenging to study antivirals, because as Johan was suggesting, really where we ought to be focusing our efforts with the antivirals is in people with earlier stage disease. And as we all know, COVID-19 really starts off as primarily a viral infection that then transitions to an inflammatory disease with lung damage and its consequences, and systemic inflammation as the main clinical hallmarks, where antiviral therapy may have less benefit. At least it's been not proven to show very much benefit, and where the at least steroids have an advantage. Paradoxically, none of the more focused anti-inflammatory drugs have shown any benefit yet. Tocilizumab, sarilumab, anakinra, now more recently canakinumab, all have failed to show benefit. And so why steroids, but not these more specific blockers of the inflammatory cascade have been successful is something we need to sort out. But combination therapy is probably the way to go in the future.
Okay.
So Peter Scher has a question to you also.
Yes, I have. Sorry, I'm a very slow writer, so that's fine. So I'm quite interested in this. You mentioned it's clear that if you want to treat the virus infection, you need to hit very early on. And then you have the kind of anti-inflammatory responses that helps the patients in a kind of with a kind of exaggerated kind of immune response. But you were touching on the topic, but it kind of intermediate phase. Normally patients come in when they feel sick, and probably they are like day 3-5 after infections. But what would be the ideal? You mentioned combination treatments, but what would be the ideal treatment to intervene in the phase between kind of the peak viremia and when you start to get the kind of host responses?
Well, I think in that early to intermediate stage, that's really, I think, where the antivirals probably have their best shot. One interesting thing about the use of corticosteroids is that they seem to have an adverse effect when used too early, and so I don't think we want to be intervening with anti-inflammatory drugs in this early stage, but really focus the efforts on antivirals. I think if testing became more widespread, especially with home testing, people can self-diagnose and then have access to antivirals, that would be an ideal circumstance in which to initiate treatment. Of course, the challenge then becomes that the majority of those individuals are not going to progress to begin with, so the number needed to treat gets to be really enormous. You treat a huge number of people to prevent adverse outcomes in a small number. You might focus that by having treatment targeted to people who are at high risk, those who are over age 65, people with obesity, people with underlying medical conditions that place them at risk for progression, to really maximize the benefit and have the best cost benefit relationship there.
So I think we could start again. My clock is 3 o'clock. So by that, I would like to introduce Peter Sjö, who is a medicinal chemist, currently discovery project leader at the Drugs for Neglected Diseases initiative in Geneva. Peter has more than 25 years of experience from the drug discovery in the pharmaceutical industry. And he has focused over the years, has been on discovery and development of novel treatments, targeting inflammatory, respiratory, and now recently infectious diseases. So we're glad that Peter is here with us today. So please share your screen, Peter.
Yes, thank you, Anna-Lena. I hope that my voice carries through. See if I can manage the technology as well. So can you all see this? Good. So I will talk about drug repurposing and a project that we have been driving to identify drug repurposing candidates for the anti-Covid study that is currently ongoing in Africa. So, but first, just 2 or 3 words around Drugs for Neglected Disease Initiative. It's a non-for-profit organization that aims to discover, develop, and providing access to a new and affordable treatment to patients in low and middle income countries. And it was founded in 2003 by the Doctors Without Borders and the World Health Organization and a group of other international research institutes. And since our start in 2003, we have delivered 8 new treatments to patients. And primarily up to now, we have been engaged in antiparasitic diseases, but also have had some development activities in the fields of HIV and HCV. And also since a couple of months, or I should say almost a year now, we have been put quite a hefty effort into SARS-CoV-2 and fighting the Covid-19 pandemic, primarily in Africa. So first, a few words around the anti-Cov consortium and the anti-Cov clinical studies that's been launched recently. So it's a large African study that is testing. The aim is to test multiple early treatment options, and with the aim to prevent disease worsening, so that people need to avoid people getting hospitalized, since hospitals and clinics is sparse in many parts of Africa. And the study is a pretty large study and will include up to 3,000 patients at 19 different clinical sites in 13 countries in Africa, and you can see them all displayed here. And this is a huge kind of collaborative effort, which includes 26 different organizations coordinated by DNDi that will conduct the studies in the different countries. And the anti-Cov, it's an open label, randomized, adaptive study. And I think that a word adaptive is very important here, because this will allow us to add and also remove treatment arms whilst the study is ongoing. So we can add new treatment and we can take away treatments that in the kind of intermediate reviews don't show good enough signals. And the aim of this study is to, as I said in the beginning, is to treat mild to moderate cases of COVID-19. So it's not a prophylactic study, and it's not aiming to treat patients which are in the severe disease states. So it's the viral symptoms, patients coming in showing viral symptoms, and also patients who have uncomplicated pneumonia, and we are measuring that by the oxygenation, which should be larger than 94%. And as mentioned, there will be a number of treatment arms in these studies, and I will primarily talk about our work leading up to the nomination of candidates for the third arm. But right now, and the clinic trial has started, and it was officially launched last Tuesday. And currently there are 175 patients in the study. And the first two treatment arms, and if we think about what's said in the previous presentation, it might sound a bit awkward that we are trialing drugs and drug combinations that in many studies have shown to give poor or limited efficacy. But in a kind of African setting, this actually makes quite a lot of sense, because both lopinavir, ritonavir, and hydroxychloroquine are drugs that are already being used locally to treat COVID-19 infections. And the study here aims to provide a definitive answer if these drugs work in a mild to moderate patient population. And many of the studies conducted so far, there is a kind of the patient segmentation causes some confusion in the interpretation of the results. But nevertheless, our task, or the work that we have been doing, has been to try to identify repurposing opportunities that could go into as a third arm in these clinical studies. And right now we have a number of drugs that we are looking into, but right now it looks like the combination of drug of a drug called nitazoxanide and atazanavir ritonavir is among the top candidates. It's not finally decided, but this is one of the kind of clear favorites that we have in our pipeline. So how did we address the challenge, and how do you identify a good repurposing candidate that fits the kind of needs of the anti-COVID study? So, and just to mention that before going into that D&I has a kind of 3-tier effort in the COVID-19 pandemic response. One is the short term, and that is support of the anti-COVID clinical trials. But also what we want to do is to support and help to build a translational understanding. How do we translate data from in vitro to in vivo to patients? And for COVID-19, it's really difficult to understand how different models are translating into finally into patient efficacy. This is something that we are working with. And then we also, as many other organizations, we also will embark or have embarked into new drug discovery, both targeting the next generation of coronaviruses, but also thinking a bit more long-term. Are there things that we can do to have compounds phase 2 or phase 3 ready at the kind of outbreak of the next pandemic? But I won't talk about that. So selecting a candidate for the anti-COVID study. So the objective that we have, as I said, it's to identify candidates for use in treatment of mild SARS-CoV-2 infections. And there are some constraints that we have applied when we started the work. One is that we clearly said at the outset, okay, since we are targeting the viremic phase of disease, we need to kind of select for drugs that are shown a kind of antiviral effect in vitro. If there would have been in vivo data, of course, we would have used that as well. But when we start the program, there were very little SARS-CoV-2 in vivo data. So we focused on the in vitro data. And also there's a clear focus on approved drugs. And we know that there's a lot of experimental treatments in different phases of development, but in order to rapidly respond to the challenge, we need to have compounds which has a well-known safety profile and preferably is available at a kind of low cost, because that's the regions that we're operating in don't afford expensive treatments. And oral route is clearly preferred. We can think of a one injection, maybe inhalation would work, but the work that we have conducted so far has solely focused on identifying oral treatments. And this has to do with access in the African countries and distribution, lack of cold chains, everything that is complicating logistics in the low-income countries. And of course, there's a number of challenges attached to this. One is that the whole kind of COVID-19 SARS-CoV-2 space, it's moving very rapidly. What you knew in March, April has changed a lot, and maybe if we had all the data that we had today, we would have made other decisions. But at each point in time, we need to make decisions based on the data that we have available at the time. Of course, we recognize the limitations, the weaknesses, and we try to address these, but this is the facts of the work that we're doing. And also, as I mentioned previously, there is a kind of lack of global consensus on the translational value of the different in vivo and in vitro assays used to study the drugs. So how did we then approach it? So we basically followed the kind of 4-tier approach, where we look on what are the available data, we evaluated the data, and then we need to some kind of prioritization. And then once having made a selection, then we moved into validation phases, try to validate as far as possible the recommendation, the selections that were made. So the data collection phase, it's kind of self-explanatory in a way, but it was collecting all available efficacy data in vitro, in vivo, or any clinical trial data that is known. And then also to understand what are the kind of currently recommended dosing, plasma protein binding, Cmaxes, where are the kind of clinical safety data, how can we treat the patients? And also what is important, I think, is to understand what is the kind of the primary pharmacology that is attached to the drug. And maybe you wouldn't go forward in a mild population with a strong antipsychotic or a very potent beta agonist or something like that. So we made that kind of early filtering to remove. And in total, we ended up with 68 drugs that we considered, okay, these are worthwhile to take a closer look at. And then to achieve the kind of ranking of these compounds, we did a very kind of basic and kind of estimate. We say, okay, what is the chance that with approved exposures of the drug in human, will we be able to cover the EC50 in vitro EC50 reported for the particular drugs? And then we set a threshold to say all compounds that on using this very simple method performs better than hydroxychloroquine. These are drugs that we should do a bit more in-depth study doing PK/PD modeling, and also start to consult with experts who have worked with these treatments previously. And from this work, we identified 14 different drug and combinations. And as mentioned before, drug combination has been kind of a cornerstone in the treatment of at least chronic viral diseases. So it's maybe not a bad idea to kind of include combinations of drug also for SARS-CoV-2 as a kind of viable option. And then the final prioritization, that was maybe we said we put the package together, and then we kind of presented this to a kind of consortium of experts from a number of different organizations, and together we scored the different options based on the preclinical evidence, the clinical evidence, and also on the available safety data. And one outcome of the initial discussion was that there was a strong preference for combinations, mainly because none of the individual drugs were seen as being kind of efficacious enough to kind of without risk moving forward. And so we basically said, okay, in order to increase the predict the kind of expectation of outcome, we said a combination is probably better, but it's still not confirmed. And on our list, first priority came the combination of atazanavir, ritonavir, and the drug nitazoxanide. I will come back to this and talk a bit more about what are these drugs and where have they been used, etc. And then to further validate the selection, we have done a lot of additional work. We have performed extensive modeling, and we also start to look more closely into the proposed dosing regimens, the dose selections, drug-drug interaction potential, and also done a bit of work trying to preclinically validate the drug and drug combinations using in vitro and in vivo data. And also, I should say that this is also building up to our ambition to increase our translational understanding, but I won't talk more about that. So what is then atazanavir and what is nitazoxanide, and what is the rationale for their use? So these drugs, they are very well-known compounds. They have been administered to millions of patients over the years, and there is, based on at least on our analysis, limited drug-drug interaction potential for these compounds. So we don't see any obvious safety concerns combining these. And then while selecting two different, the benefit is of course that you're combining two different mechanisms of action, and thereby you hopefully can expect to have a better efficacy in the end. So atazanavir, ritonavir, it's a well-known HIV treatment. It's an HIV protease, HIV-1 protease inhibitor, and there are some evidence supporting that the mode of action in SARS-CoV-2 happens through inhibition of the main protease, and this has been, I wouldn't say fully kind of confirmed, but there are some experimental evidence that this actually is the case. It's a kind of medium potency inhibitor of SARS-CoV-2. In the Vero cell system, it's the data reported between 0.5 and 10 micromolar, and in lung epithelial cell lines, the efficacy is reported to be 0.6 micromolar. So they are kind of intermediate potency, I would say. Nitazoxanide it's a kind of broad spectrum anti-infective, which has been used in treatments of parasites like Cryptosporidium. It has also been proposed as treatment of tuberculosis, but this, if I understand correctly, these studies has not been successful. And also it has been trialed in both for influenza virus and other virus infections and show limited efficacy. So, and it has a broad spectrum mode of action. And I haven't really seen any kind of firm evidence, but for SARS-CoV-2, but for influenza viruses, it's reported that the nitazoxanide blocks the viral hemagglutinin and thereby the replication and maturation of viruses. And there's also reports on inhibition of the polymerase complex, and it also affects through host effects uptake and transmission of the viruses. It has also intermediate potency. Nitazoxanide itself has 2 micromolar, but for these compounds, it's important to understand that the nitazoxanide itself is not active ingredient is actually its primary metabolite, tisazoxanide, which also is an active species, at least when tested in the SARS-CoV-2 aerosol systems. Importantly for these drugs, when we do the modeling, we can see that there's a clear preference for atazanavir to accumulate in the lung, and that's due to its slightly basic character. So there is an accumulation, predicted accumulation in the lung, which is between 40 to 80-fold higher than the reported IC90 value, in vitro IC90 value for SARS-CoV-2. For nitazoxanide, it's the same. It's kind of reports higher Cmax, which covers the EC50s. Also the Cmin values covers the EC50. So it's kind of a fair selection, and the pharmacology and pharmacokinetics looks reasonable for these compounds. So how to select the dose? So of course there's a recommended dose for these compounds, and it's in its current treatment in HIV. Atazanavir is a daily dose of 300 milligrams, and with the standard treatment, ritonavir, it's an antiviral, but it's added to boost the PK of atazanavir in the kind of drug treatment. So that's why ritonavir is in this mix. And if you give this once a day to patients, you can see that on the right hand panel that you have a kind of slow increase of the concentrations up to a steady state after approximately 5 days, which in this case is not so good because you want to hit the virus infection early on. So for atazanavir, we propose that you have a loading dose. So you give on day 1, you give atazanavir, ritonavir 2 times a day, and then you continue with the once daily dosing. And you can see here that when you look on the predicted exposure in man, within 24 hours you reach more or less the steady state concentration, which in this case would be beneficial to address the kind of virus infection early on. So for nitazoxanide, the situation is a bit more complicated, and we are actually proposing a higher dose than the recommended one. So we're proposing a 2 grams a day dose for 14 days compared to the 1 gram a day given 2 times 500 milligrams. And this is because the simulations that we are doing, it shows that if you use the kind of recommended 500 BID dosing, only 1% of the patients would achieve EC90 at C trough. Whilst if you double the dose, 1000 milligrams 2 times daily, then 48% of the patients would achieve EC90 at C trough, which is quite significant. We have done the model. If you go to 1500 milligrams 2 times a day, then you cover 100% of the patients. But probably because of the third bullet here, the safety concerns, when you increase the dosing, you start to see GI-related effects, and also you start to see accumulation of tisazoxanide, which is the primary metabolite, and it's glucuronide. It's seen after 7 days. So we think that 1000 milligrams will provide you kind of a decent coverage, and there is also now clinical support for this from a study in Brazil on nitazoxanide. I will come back to that. So, but how do we manage the safety concerns? We when you look on other studies performed, it's other people have gone up to higher doses, and there are some side effects, but they are not really significant if it's not severely diseased patients, like if you have tuberculosis patients which are severely infected. That's what we have been able to find out. So we can see that, for example, that if you give 500 milligrams 3 times a day, as they done in this Brazilian SARS-CoV-2 study, where you see effect on day 7 on on the kind of virus levels, then it's well tolerated. You can dose up to 4 times single dose without seeing significant side effects. And also you can give the drug 500 BID for extended periods of time without kind of any kind of significant side effects. So we are fairly confident that 1000 milligrams BID will be acceptable in the patient population that we are targeting. So how do we do the validation? So since we did the selection, we have done quite a lot of work on this. We have looked into, we have data from the mouse model, which I should say earlier up front, it's not the perfect model for SARS-CoV-2 because the mouse clears the viremia very quickly, so it's difficult to get significant effects. But we do see for both nitazoxanide and atazanavir-ritonavir a reduction of the peak viremia of more than 1 log unit. And also for nitazoxanide, as I mentioned, we do have positive effects on the viral loads in a clinical study in Brazil where they dose 500 milligrams 3 times a day. I should say that this is not an optimal dose. It's actually only covers 28% of the patients at EC90. And for atazanavir, we don't have any experimental data beyond the mouse at this point, but the extended modeling that we have done still suggests that this is a good choice to move forward with. So, and we are continuing to do the validation work. So we have hamster studies ongoing where we are evaluating the results for the drug, individual drugs and the drug combinations. We have done PK studies. What we want to do in these models is to match the human dose in the hamsters, and this maybe has been very difficult because it's really difficult to get exposure enough in the hamsters to match the predicted human exposure, and that is very much kind of practical reasons. It's really difficult in a kind of BSL-3 lab to do more than 2 times a day dosing of compounds. We can't kind of increase the dosing frequency, and it's the kind of volumes injected or prevents us from giving more drugs at the same time. And so we are doing this and we are evaluating this, and then we are continuing to evaluate ongoing clinical studies like the Nigeria studies, and there's also studies in South Africa ongoing with nitazoxanide that we're closely monitoring these studies. And once we get the results, we bring that into our kind of PK modeling to analyze the outcomes, to understand the results a bit better, like we have done for the Brazilian study. So, and right now we are in the preparation of the final recommendations of atazanavir-ritonavir and nitazoxanide as to be moved into the anti-COVID studies. So, and that's actually all I want to say. But before ending, I just wanted to thank both our collaborators at the University of Liverpool who have done a lot of the modeling, and also to my colleagues at the NDI and our kind of consultants and other collaborators. And also we do receive funding to do the work, and MSF and UK and the Swiss Agency for Development and Cooperation has been kind of key of supporting the NDI overall, and BMBF in Germany has been providing us funds both for the anti-COVID study and also for the discovery work that we are doing. So thank you for your attention, and I'm happy to take any questions.
So thanks a lot, Peter, for this very interesting presentation. And please write your questions in the chat, and maybe I can start. So you have been, I think we heard it before, that combinations of drug is very important. And I wonder, is it possible, you think, to screen for finding new combinations screening in cell models or other sort of early?
Yeah, no, it's been done, and we have some data from NIH where they actually done kind of in vitro combination screening. So it's definitely possible, but of course it's a lot of work doing this. But what I should say, I think personally, my view is that there is a difference between kind of targeting chronic virus infections like HIV and HCV, where you have the risk for mutations, that there is another driver for maintaining a kind of high drug pressure, is because you want to kind of avoid mutations. That's I don't think it's really the same kind of pressure for the SARS-CoV-2. It's an acute infection, and it's kind of, in most people, it clears by itself. Yeah, but in the repurposing, I think it has a room and a place, because you are, as you've seen, we are dealing with imperfect drugs. They are, in best cases, intermediate potency. And so in order to kind of get the maximum pressure for a short time, it's probably good. You can use combinations to compensate for the kind of lack of efficacy for the individual drug components. That's how I personally see it.
So I see there is a question to Peter Mario. Or should I read the question, Mario? So what kind of in vitro COV-2 efficacy data did you use during the candidate selection?
So we used published data and try to kind of do kind of an assessment of this data, and then of course we have followed up doing our own in vitro studies later on. But when we did the selection of the compounds, we used the published data. And of course, there's for some compounds, you can say that the published data, they are kind of fairly coherent, but for other compounds like favipiravir that was mentioned before, you have a really broad range of different data. So you need to use the published data with cautious, and also be aware of what is the type of assay that has been used. Is it, in most cases, it's different Verocell systems. Preferably, I think we would all like to use lung epithelial cells or cell lines, but these data are pretty sparse, I would say.
So we have another question from Christian Sandberg. Based on available data, can you predict any concerns with regard to drug-drug interactions using the combo nitazoxanide and...
Yes, we have looked into this. Atazanavir is a UGT transferase inhibitor, so it's kind of theoretically this could lead to that you accumulate since the kind of tisoxanide, which is the kind of primary metabolite of nitazoxanide, it's glucuronidated, and inhibiting UGTs that could theoretically lead that we get an accumulation of tisoxanide in systemically. So that's one of the things that we've been looking into. But fortunately, for many of these drugs, all the kind of data is available, so you can do your simulations. And when we've done the simulations on UGT 1, 2, and using UGT 1, 2, and 3, we can't see that there is a significant. We don't see an effect based on the inhibition of the glucuronidation on the tisoxanide levels. So that's yes.
Okay, so there's another question here from Päivi Ostling. As our knowledge increases, how can the efficacy of in vitro data and the whole prioritization process, how could this be done also for new screening data?
I think the big advantage is that when you are kind of working with new compounds, I think inherently they will be much more potent. So you are maybe already from start a 10-fold higher in potency. So you you will have much clearer readouts in your models, and so I think that's I don't know if that's a good answer. I think the key thing for us, and where we really have struggled, is to kind of understand the data from different models, in vitro, in vivo. How can these be used? Can they be used to kind of rank compounds against each other? Can they be used to kind of understand the efficacy in patients or in the animal models? And I think the more we learn, the more we will learn which are the models that actually can have a predictive value to a kind of the higher system, if you say, and finally to patients. And I think it's the same with, if you take the hamster model, is very popular now, but okay, for different mode of actions, how should you time when you do your sampling, etc., because it's not necessary that you will see the effects on day 1, maybe you see it on day 2 or 3 or 4, or so this is what I think we will need to learn. Yeah.
Good. So we have a final question from Jens Karlsson. Please speak, Jens.
Hello.
Yeah.
So I have a question. So repurposing is a brilliant idea, of course, but with all the efforts ongoing, is there a point where we should assume that if there was something in this small set of compounds we could use, we should have found it?
Yeah, I fully agree with you. I think we are approaching that point. And I think that the main reason for doing drug repurposing of compounds is another thing, but drug repurposing is that it provides you a shortcut to enter into patients, because you have all the safety data available, so you can understand kind of your kind of maximum doses in relation to your kind of predicted C trough values, for example. So it's just a kind of shortcut to get quickly into patients, and then you carry a lot of weaknesses with the compounds that you have selected, and mainly it's related to lower efficacy. I think that's the key thing. So of course, when there's newer and more efficacious compounds on the way, it kind of, then the interest for repurposing should go down. And also I think for the COVID, I kind of doubt myself that any new drug discovery will have an impact on the current epidemic. So I think it's more the pandemic preparedness that is the important thing. And I think there is also a learning moving forward. If you can develop a vaccine in 1 year's time, as it happened now, then it doesn't make sense to have something which is in phase 2, because it will still take quite some time. So you need to have something that available that can go into phase 3 study, more or less. Or yeah, so it's there are some learnings from what's happening, I think.
Yeah. So thank you. And I think we will have more comments from Johan Knights on repurposing and the novel drug development. So we can continue the discussion in the as well during the panel. So by that, I think we have 2 minutes bio break, and I hand over to Johan. Thank you.
Thank you. So I will share my slides in the meantime.
Okay, let's start. So I'm very happy and honored to present Johan Knights. He's a professor at the University of Leuven in Belgium, and is fully focused on development of novel antiviral strategies against a number of viruses. And his laboratory has also a long-standing expertise with respect to development of small animal models. He has published more than 500 papers in drug discovery antiviral, and he's currently this year a member of the Belgium National National Advisory Board on COVID-19. And he's going to present some exciting publications. This is recently published. And I just want to say that I'm very happy that you're here. I had nice talks with you at the meetings we met 3 years ago in Smögen. And you had an excellent presentation there. And then later on in San Rafael last year, you had an excellent presentation. I had nice chats and discussions after that. So I'm very happy you're here. The word is yours now, Johan.
Thank you, Johan, and I'm happy as well. Thank you for the kind invitation. This has been really an exciting meeting, and I think that the previous speakers did a really good job. And I think it's nice to sort of come after them, to put what we are doing a little bit in the context of what they have been saying. So the only words I know in Swedish is hur mår du? And if at the time that we could still travel, I could say åt du frukost på planet, but that's all I can. And I as you know, Johan, and some other people on the call, I love Sweden. My daughter also did her Erasmus in Umeå, so it's a nice country. So too bad I cannot join you in Uppsala today. Perhaps next time if we get the problem solved. So what I would like to do is to give you a bit of a view on the use of animal models to develop antiviral strategies and also a vaccine approach. You can follow us on Twitter, my own Twitter account, and that one of the lab. And this is the website of the lab, so it's easy to find us. Well, as you can see on the slide, in September 2019, I was in Wuhan at an international virology conference. And I mean, this is not me here on the stage, that's the organizer. And you see here a picture that I took of Wuhan. So it's a huge city, 11 million people. It's a mix of a modern and a historic city. And this is the Yangtze River, so we made a cruise on the Yangtze River. I could never have imagined at the time that just a couple of months later that would be the epicenter of the next pandemic. And actually, quite likely in September, the virus may have been starting to circulate already at the time that I was basically on the stage explaining that at this conference, that the world is not prepared for the next epidemic pandemic, and that we would need broad-acting antivirals to stop a new virus if it is emerging. And I gave the example of a couple of viral families, including Paramyxos, Bunyas, but also Coronas. So it's sort of, it's kind of bitter that that I was explaining that at the time that the virus started likely to circulate. So I will talk about the use of animal models, but first in the context of antivirals. As you know, what we have in terms of antiviral therapy are drugs to treat herpes infections. For example, acyclovir, but others as well, to treat HIV. And I think it's quite remarkable that we can, I mean, although there is 37 years after HIV has been discovered, there is not yet a vaccine, but we do have these cocktails of ultra-potent drugs, and people with HIV, when it was at the time a deadly disease, and I think we all remember, oh, my slides are moving by themselves, that at the time it was still a deadly disease, that we can now just treat patients with just one pill a day, and that shows what really potent antiviral drugs can do. So not just repurposing drugs, but really highly potent antivirals with nanomolar potency. Same thing for hepatitis C. I think this is a revolution, what has happened in hepatitis C field. Hepatitis C can be cured today. People with chronic hepatitis that have a high risk of developing liver cirrhosis and liver cancer can today, with just 2 months to 3 months of 1 or 2 pills a day, can be cured from this massive hepatitis C infection. And by the way, for HIV and also hepatitis B, tenofovir, the nucleoside phosphonate, has been discovered here in our lab by my mentor Erik de Klerk at the time, and is so the cornerstone of, for example, these combinations, TRIBUILT and other combinations, is a cornerstone and one of the most widely used anti-HIV and anti-hepatitis B drugs around the world. And also we have antivirals against Tamiflu. I just show here a couple of examples, of course, of drugs available. And I think we all agree that we would need even more potent flu drugs. But this is what we have, and we do not have antivirals against RSV and other paramyxoviruses. We do not have drugs against enteroviruses, also not against rhinos, not against adenoparvoarena. Bunya's I label now corona, but this is a slide that I show since years. Not against flavis and others. By the way, against flavis we developed a ultra potent, and this is really ultra potent, means picomolar potency, against and pan-serotype dengue drug that is currently in clinical development at the Janssen Pharmaceutica. But the point I would like to make is that we do not have, against many viruses and many viral families, we do not have drugs available. And so this is here a screenshot of a TED lecture of Bill Gates 5 years ago, and he said, if anything kills over 10 million people in the next few decades, it's most likely to be a highly infectious virus rather than a war. And we're not ready for the next epidemic. And I think he was right, although he's not a virologist. And so my point is, and I explained that also in Wuhan, imagine that we had, back in 2019, 2018, 2019, that we had broad-acting pan-corona antivirals, and we knew 4 coronas that cause mild disease and 2 MERS and SARS-CoV-1 that cause severe disease. So we had human coronas against which we could develop antivirals. And just imagine that there had been an investment made to develop such an antiviral towards phase 1, demonstrating safety, and perhaps with some of the mild coronas show that this is also effective against infection. Then there could be a stockpile, and this could, for example, be with the WHO. And then just imagine that at the time of the outbreak in Wuhan, at the start of the outbreak, that together with that these drugs could be shipped to Wuhan, and that the first patients could be treated, and that the doctors and nurses could be treated prophylactically, and contacts of these first patients could be treated prophylactically. Then perhaps you could have stopped this virus from spreading. You could have bought time to further work on vaccines, but it didn't happen. The world is buying military equipment for countries to protect them against other countries, but we're not protecting ourselves against these nano weapons, these viruses. And I think we should do it. And I think this should go hand in hand with new diagnostic tools that allow to do very rapidly, if new pathogen is emerging, to very rapidly do mass testing. I think the classical old PCR assays, as we're seeing now, I think this will be outdated. We should go to more fancy methods. That's possible. Mankind put in 1969, we put people on the moon, and today we're still not able to stop a new virus if it is emerging. So I think we have some work to do. So there's some future for virologists. Remdesivir has been nicely reviewed, so I don't have to comment on that. But you know that remdesivir is targeting the RNA-dependent RNA polymerase. We know from HIV and hepatitis C and others that the polymerase is, of course, a good target. There's more that can be developed than just RNA polymerase inhibitors. And we also heard earlier about the protease as a good target, and that's shown for HIV, and something is happening with my slides. That for HIV and hepatitis C, that the protease is a good target, and also in my lab, even before 2020, we worked on the protease as a target, and we have some nice compounds. So target-based drug discovery, we do that, we like it, but what I like even more is phenotypic screening. And I call it basically looking for the needle in the haystack. And the haystack is the infected cell. the black box almost of the infected cell, where a lot of interactions happen also between the virus and the host cell that you can't put into a target-based screen. So this is why we do a lot of phenotypic screens as well. You see here a part of a 384 well plate in which we seed cells that are labeled green with GFP. And if we then add the virus to it, after 4 to 5 days, the cells are being killed. And if we add, for example, here remdesivir in dilutions, you see that the cells are protected against the CPE. And so we just put this into high content imager and can do screening. But of course, you don't want your technicians to sit there day and night, certainly not in a pandemic situation, to do pipetting and testing. So a couple of years ago, we thought that we should be able to build a fully automated lab, a high biosafety fully automated lab. And here you see a drawing of what it basically is. It's in blue. It's a completely airtight box. With in gray, you see the incubators. We have 4 incubators that can be accessed from the inside. You will see a little video in a minute. On top of that is all the air handling. And this is a glove box system attached to it. So the whole box is about 3 on 3 meters. And this glove box system is giving access to a walk-in high biosafety lab where we can then basically culture the virus and put it into the system. You can, on my website, there's a video, and I will show a short fragment of the video here. So you here see the corner of the lab with 2 high content images with the blue lid. And now on the, this is located in our biosafety lab. And here you see an inside of the facility. Here you see in the 2 high content images here at the left-hand side, you see the central robot arm that is taking the plates and putting them at different positions. For example, here at the barcode reader. And then the central arm in the background, you see an arm that is deleting plates. The arm goes to the high content imager that takes a plate and brings the plate to different position. This is how the system is built up. Basically, every piece of equipment, except for the central robot arm, is in duplicate, so that if one instrument would fail, that we still have a backup. Here you see that the robot is bringing, well, one of the robot arms is bringing the plate to one of the 4 incubators. And after 5 days, the software knows that the plate is ready and has to be taken out and has to be put, for example, in the high content image to do readout. So this system is working 24 hours a day, 7 days a week. And while our people are sleeping also. And as you can imagine, this is increasing our the throughput very much. And so far we screened 1.7 million compounds against SARS-CoV-2. Not only our own compounds, but also we do that for academic chemists around the world that ship us compounds. We do it for biotech. We do it for almost all big pharma companies that are shipping their compounds to us, and we then run the screen. Okay, but I promised to talk about animal models. So I will explain what we do about that, and mostly the hamster model. First of all, at the time that we had access to the virus, we tried first in mice, and I put this here so that you can see better my slides. And we just published that in Nature Communications about 2 weeks ago. But mice are not a good model, as you know, or may know by now, but we used bulb C mice, and we used black 6 mice, and you know, bulb C mice, there's also skid mice are bulb C derived, and black 6 have, based on black 6, we have if mice and interferon lambda knockout mice. The bottom line is, if you just, so this panel B is wild type and skid bulb C, and you see that if you inoculate the virus in the nose, that is just 1 to 2 log increase of virus in the lung. It's not sufficient. Also in skid mice, to our surprise, we did not see much. Also in black 6 mice and wild type in blue, there was not much. In if mice, perhaps it's a bit more, but we still did not find that this was sufficient. Likewise, well, this is timing. Likewise, if you look at the gene expression map, you see that in SARS-CoV-2 infected animals, there's a bit of an increase in some gene expression. That's for the IF9 mice, but nothing spectacular. Also, when we look in panel D at the lung pathology for BALB/c mice, for SCID, and in the Black 6 mice, also in the IF9s, there is a bit of pathology, but just not sufficient if you want to test antivirals, if you want to test vaccines. So we quickly realized that, and we moved on to hamsters, and we were lucky that we had not only wild-type hamsters, but also STAT2 knockout hamsters and interferon lambda knockout hamsters. So what you see here is I should say that we use a passage 4 virus in the studies and also passage 6. The passage 6 is less pathogenic in hamsters because it has a mutation in the furin cleavage site. All right, so in panel B, you see in these hamsters that we have high titers of viral RNA in the lung, but let's have a look at panel C to infectious viral titers. So also there we have pretty high titers of infectious virus, but in the STAT2 knockout, there is about 1.5 to 2 log increase in infectious viral titers in the lungs. Interferon lambda knockout hamsters, there you don't see that much of a difference with the wild types. Now, if you look at lung pathology, you see right away in panel D that in STAT2 knockout hamsters, we do see much less pathology than in the wild types, which is in contrast to the fact that you see high titers of virus in the lungs. Now, in panel E, if you now look to viral RNA in blood, you see that there is not much in the wild type hamsters, but we do see a lot of virus, so huge dissemination into the circulation in the STAT2 knockouts. Again, interferon lambda knockouts, there we do not see much of a difference. And likewise, if you look in the spleen, the intestine, the liver, you see that each time that in STAT2 knockouts that we have a huge load of virus and not in the wild type animals. When we look at more closely at the pathology, this is a COVID-like pathology, as you can see in panel A. Panel B is a healthy hamster. What we see is a bronchiolitis, necrotizing bronchiolitis, perivascular edema, clearly also bronchopneumonia, perivascular inflammation, peribronchial inflammation, and also apoptotic bodies in the bronchus wall. So quite a pathology, and certainly in those hamsters that have been inoculated with a low inoculum, an early passage inoculum. Strange enough, despite the fact that these hamsters have such a bad lung disease, they don't look very sick, they don't lose much body weight, and they recover basically by day 8, day 9, most of them have recovered. Again, here you see in panel C, the histopath score. So again, in STAT2 knockouts, you see it's much less than in the wild types. Okay, just also here, so you see lung pathology much less in the wild type. And if you just in panel B look at the matching of the histopath with infectious virus titer in the lungs, just look at the wild types and just, don't look at the different data points, but just look at the gray bars. You see that at the left-hand side, infectious titers, at the right-hand side, y-axis, histopath score, it's not much of difference, but in STAT2 knockouts, it's really a serious difference. We also use micro-CT to assess and evaluate basically over the different time points how lung disease evolves. You see that in the wild types, we do have a very much affected CT scan, which is not the case in STAT2 knockout in infected hamsters, despite that the virus is all over in the lungs. You see that also in panel D and panel E that the micro-CT, if you quantify that, is sort of normalized almost in the STAT2 knockout hamsters. Also in the gene expression heat map, you see that the STAT2 knockouts, in contrast to the wild types, that there's not much of an increase. And so basically the conclusion is that, and that's in this Nature Communication paper that we published, that STAT2 knockout signaling restricts dissemination of SARS-CoV-2 in hamsters, but is driving basically severe pneumonia in these hamsters. So to put it in kind of a graphic format, in wild-type hamsters, we do have a lot of pathology, and quite a bit of replication. We do not see any dissemination. In STAT2 knockout hamsters, there's almost no disease in the lungs, whereas there's massive replication in the lungs, and there's also dissemination of the virus to the rest of the body. And you can also see this in the spider diagram, that in red here is a STAT2 knockout hamsters. We have huge replication of virus, but very little pathology, as you can see. So not much bronchopneumonia and not much inflammation, for example. So yeah, quite an intriguing observation. Okay, but we want next to use the model to assess the impact of antivirals. And so we looked, for example, at the kinetics, and you can see this on the y-axis is the infectious viral titers. You can see by day 1 it's going up, and by day 4 we reach already a plateau. And again, we can look at the CT scans, and we can look at the histopath, and of course we can then look at the impact of drugs. And so what we one of the compounds that we wanted to test is Avigan. You may know that compound, of favipiravir, as you can read here. It's a Japanese anti-flu compound, and we did find some activity in cell culture, but nothing spectacular. But we still wanted to test it in hamsters. And we did that in parallel with hydroxychloroquine and also azithromycin, because, as you know, some have claimed that the combination of hydroxychloroquine and azithromycin may be beneficial. By the way, we published that just recently in Proceedings of the National Academy of Science of the USA and PNAS. All right, so what do we see in hamsters? So here we look on the y-axis at infectious viral titers. So we infect hamsters intranasally, and we start treatment, and we continue treatment for 3 days, and then at day 4, at the peak of viral load in the lungs, we look at different aspects, including the infectious titers. In red is the high dose of favipiravir. I know it's high, but you can see that in gray are the titers in the untreated animals, and you see that we basically bring, in most of the animals, by giving this high dose of favipiravir, we bring the infectious titers down to undetectable. With lower doses of favipiravir, there is much less of an effect. Hydroxychloroquine, here in blue, has no effect. Hydroxychloroquine, when combined with azithromycin, has no effect. So when we imagine that we had early on in the pandemic that we had this hamster model, but of course it takes time to establish this, we could have told the clinicians that they should not go and test hydroxychloroquine, because actually it has likely no potential. This is the effect on the body weight. So this high dose of favipiravir is quite well tolerated. And we look in panel E at the lung score. So in red, you see that the treatment with favipiravir is also reducing the pathology. And also in the lower graph, you see that favipiravir at high dose, so here the 1000 milligram per kilogram at high dose, that it also cleans up the lungs. At the lower dose, this is not the case. And also hydroxychloroquine, you do see that there is lung disease, as you can see from these CT scans. Question is this achievable in patients? We believe yes, because when you look at the PK, you see that we have exposures of about, so this is a plasma trough, about 7 microgram per ml. And we know that from the Ebola studies that this can be achieved in patients that receive high doses of favipiravir. So this is interesting. Now, an experiment that I like very much, which is also in the publication, is a transmission experiment. So what we do is we put an infected hamster together with an uninfected animal. And then it's like putting 2 people, an infected people and an uninfected people, in the same pub or the same restaurant. You have transmission. You have always transmission. We see it all the time. Now, if we treat the sentinel prophylactically, and then 4 days after you put them together, look in the lungs of the sentinels. And what you see then, so first look at the gray. So in the index hamsters all have a lot of infectious viral virus in the lung. But all the sentinels, and of course it's only 2 mice per, 2 hamsters per box, so you have to, this is pairwise, but you see that all the sentinels become infected. Some have huge infectious viral titer in the lung and some have low titers. But look at favipiravir, you see that all prophylactically treated hamsters are completely protected against the infection. Hydroxychloroquine, you have to compare the open blue circles with the open gray circles, has no effect. So this is interesting, we believe, because it may, although favipiravir is not a potent compound, not at all, but it may open some options for prophylactic treatment. For example, in long-term care facilities, where you have a few people that come down with the infection, could then give prophylactically to the others this drug. And actually, we will start here in Belgium a study, an ambulatory study in elderly facilities, long-term care facilities, to prophylactically, well, actually treat people right at the time that they have received the diagnosis. The hamster model is also ideally suited to test antibodies. We heard also a nice summary on the use of antibodies. Here we work with a team at the University of Ghent and Jason McClellan, also in the US, using these nanobodies from alpacas. This is just one experiment. We have many more and even more spectacular data, but you can see that even with one of the not too potent nanobodies, that we have about 5-log reduction with just one shot of the antibody in the hamster, so pointing towards the potency of such antibodies. With some of the other antibodies, we achieve complete sterilization. Also here with the team of Davide Corte, Katja Fink, and also David Wiesler, with the antibodies from Vir and Humaps in hamsters, we do see that we can achieve complete sterilization with just one injection with low amounts of the antibody, just 1 milligram per kilogram of a cocktail, for example. So quite promising, at least in the animals. I would like to then shift to what we do on the vaccine, and I'm happy to tell you. So you sort of the first to hear this after the publication that came out today. So we published our findings in Nature. It was published online at 11 a.m. And so what we do is we use the yellow fever vaccine as a vector. So you know that adenoviruses have been used, for example, as a vector, but we thought, and we have been working on that for quite a couple of years, that the yellow fever is a good vector. You know, yellow fever vaccine, it's around since 1938. Max Tyler got the Nobel Prize for that. It has been given to 800 million people. One shot gives, within 10 days, lifelong protection. So if you travel, for example, to Africa or Latin America, where yellow fever is endemic, you just get your shot, and after 2 weeks, you can safely travel. Okay, what did we do? So we inserted, so in panel A, you see in white the sequence of the yellow fever genetic, of the genetic code of yellow fever 17D, the vaccine strain live attenuated virus. And we inserted the spike of SARS-CoV-2. And we basically made 8 constructs, but I just showed 3. In blue, as you can see it here, is the post-fusion variant. In green is the pre-fusion variant, where we mutate the cleavage site. And here is in red is 1. You see the plaques for yellow fever 17D, and you also see it for the different constructs. You see that for the constructs that the plaques are smaller than that for yellow fever 17D, and that also translates in the fact, I'll show you that in a minute, that this SARS-CoV-2 vaccine candidate is also attenuated in terms of safety as compared to the commercial yellow fever vaccine. Okay, we have a lot of data in the Nature paper. I invite you to check the paper if you would be interested. I just show you some data. So here, for example, we vaccinated the hamsters with 10 to the 3rd to 10 to the 4th per view of our vaccine, and then waited for 3 weeks, and then infected the animals intranasally, and after 4 days checked the lungs. Now what you can see, this is here in green with the green triangles. If you compare that to the white open circles, you see that we have with this one-shot vaccine with 10 to the 4th that we have complete protection. So 5 to 6 log reduction in infectious titers. One of the animals even reaches almost 10 to the 7th reduction. Even if we have, and this is what you see here, even if we give the hamster just one shot of 10 to the 4th and do the inoculation 10 days after infection, you can see that here in the squares that 5 out of the 8 hamsters that we do not find infectious virus back in the lungs. And 2 of the hamsters, it's nice reduction, and 1, it's 10 days is apparently just a little bit too fast. So this is, we believe, quite spectacular. We haven't seen any of the other vaccine candidates tested in the hamster model that has given this clear protection. Another pretty cool experiment, not yet published, is also this transmission model. So again, we put infected animals with sentinels, and then you see that in the non-vaccinated sentinels, they all get infected. On the y-axis, this is on a log scale. When we vaccinate the sentinels before we put them in the cage, you see that there's basically almost no viral RNA into the lungs of the sentinels. And of course, if you vaccinate the index hamsters and the sentinels, there's also complete protection. And that's also, to the best of our knowledge, the first study showing that a vaccine is protecting against transmission. What in non-human primates? In macaques, we gave them one shot of the vaccine, and at the time that we submitted the application to the ethical committee, we were not sure that we should give 1 or 2 doses, and so to be sure we gave at day 7 another shot, but we believe that this is not needed anymore. And then at day 14, day 21, we look at we look at non-neutralizing antibodies. As you can see in panel B, by day 7 we have already, and so this is just before the second shot, at day 7 and 2 out of the 5 of the 6 animals, we do have already neutralizing antibodies coming, and by day 14 we have high titers of neutralizing antibodies, which is basically at the high end. So of the other vaccine candidates, few reach those titers. Interestingly, if we measure neutralizing antibody titers against the yellow fever 17D, you see that by days 7, so after one shot, 5 out of 6 animals have neutralizing antibodies. By day 14 and day 21, they have huge titers of neutralizing antibodies. So this basically shows that this is also dual vaccine. It shows it induces immunity against both SARS-CoV-2 and yellow fever. And I'll come back to that in a minute. What is not on this slide, but this in the paper, is that if we then challenge the animals in the nose and the throat, that we do not find, except for one single data point, the virus back in the throat of these vaccinated animals. Unfortunately, not all the placebos turned out positive, but I think that you will agree that the titers that we reach in these animals are quite high and should protect against disease. I told you that we believe that this will be a safe vaccine, and it's also in the paper and there's more in the paper than what I show here, but we did a deep dive into the potential safety. Now, we used AG129 mice, which are interferon alpha beta gamma receptor knockout mice, and they're very susceptible to yellow fever 17D, to the vaccine itself. So look at this with the triangle, just one peer view of the vaccine is killing these mice after about 16, 17 days. Now, when we give our construct here in green, even 10 to the 4th peer view, just 1 out of 8 animals was dying in the end. When we give 10 to the 3rd peer view, they all survived, they're all happy. So in terms of safety in this interferon knockout mice, our vaccine candidate is at least 10,000-fold attenuated in terms of safety as compared to the commercial yellow fever vaccine. Also, when we look into STAT2 knockout hamsters, so STAT2 knockout hamsters, as you can see in the gray, black, in the gray squares, they are very sensitive to yellow fever 17D. They die because of 17D. When we in green infect these STAT2 knockout hamsters with our yellow fever SARS-CoV-2 vaccine construct. There's just one animal that had to be euthanized. So also here in this STAT2 knockout hamsters, there's a huge difference. And then, as I said, I mean, we do see antibodies against yellow fever as well. So the question was, would this vaccine protect as well against yellow fever challenge? Now, at the time we did the studies, we did not have wild type yellow fever in-house. Now we do have it. But the model that we used was to inject the yellow fever 17D vaccine right into the brain of mice. And then they develop a neurotropic disease and they die. You can see here with open circles that the body weight is going rapidly down and they have to be euthanized. Now, if we vaccinate animals 3 weeks earlier with just 400 PV of the vaccine, you see that they all protected, they just grow normally and they don't die. So this is definitely a bonus. I think this vaccine is protecting against yellow fever challenge. And this may be indeed a bonus because the WHO has a strategy to eliminate, the I strategy to eliminate yellow fever epidemics by 2026. Now, having a SARS-CoV-2 vaccine that also protects against yellow fever may definitely be a help there. Now, also in Asia, there's a risk and there's a fear that one or the other day yellow fever may emerge, may cause epidemics, because the Aedes mosquito is also endemic in Asia, in tropic places, subtropical places in Asia, and it has been shown 2 weeks ago that that these mosquitoes are also able to transmit experimentally, transmit yellow fever. So just imagine that we could, by using our vaccine also in Asia, could lay a basis, start to vaccinate people against yellow fever. This would be definitely beneficial if there would ever be an outbreak with yellow fever in Asia. So there's much more data in the Nature paper, but I hope that I have convinced you that the hamster model, and just go back to the hamsters, that the hamster model is a really nice model to assess efficacy of drugs and of vaccines. And we are, of course, we are assessing, and also for the NDI, the effect of combinations, first in cell culture, also in air liquid interface human epithelial cell, lung cell culture models. So we look at the effect of combinations. We look at the effect of combinations in hamsters to guide further clinical development. And besides sort of testing repurposing drugs, as explained, we are very active in trying to develop pan-corona inhibitors, because we do not need only corona SARS-CoV-2 inhibitors, but we will need also for the next years and decennia to come. I think it would be good to have drugs that are broadly acting against coronas, because just imagine that within 10 or 15 years we have a new pandemic strain of corona, of not the corona. It would be pretty sad that we would not that we would have developed, for example, a drug or drugs acting against SARS-CoV-2, but that would not be active against other members of the coronavirus family. My point is, and I would like to come back to what Bill Gates said, we're not ready for the next pandemic, is that we should not only have broad-acting antivirals against coronaviruses, but also against some of the other families from which we know that new pathogens may emerge. Just think about Bunya's, just think about Flavis, just think about paramyxoviruses, and perhaps, I mean, who knows? I don't want to be a doom thinking, but who knows? I mean, imagine that we had a corona emerging that, or a paramyxo emerging that doesn't kill mostly old people, but that would kill young infants, that would have a mortality of, let's say, 5 to 10%, that would have a R not of 2 or 3, but of 15 or 18, like the measles. I think we would be in pretty bad shape, and would not, if that would happen, we would not have the time even to wait for a vaccine to come, even if it comes at warp speed within 1 year. So in that situation, we would need antivirals that you could use to stop the epidemic, the starting epidemic right from the beginning. And so this brings me to the end of my talk. I have to thank a lot of people, and I have to to thank also the Gates Foundation. We are being funded also by the EU, by the Flanders Fund for Scientific Research, by the university. And I thank you all for listening. Thank you.
Very impressive, Johan Neutz. You went a little over time, but that's no problem. It was very interesting.
My apologies. So then we shorten the questioning.
That was like, the lab was fantastic. The BCL3 lab you have, the hamster model, and also to top it off, the vaccine. Very impressive. So there's hardly any questions left, but I'm asking you, what is your personal most promising drug target in Corona, in the Covid-19, the SARS-CoV-2? What do you think? And also for respect of broad spectrum coronavirus drugs.
For now, what we have, I think the most promising is the protease. We have already we work with some teams on protease inhibitors, as said. So drug really structure-based drug discovery, and we are down already to let's say low nanomolar potency drugs, drug candidates. And so in the HTS, the high throughput screening, of course, we pick up hit compounds that we then optimize with the medicinal chemist, because we do, of course, work a lot with medicinal chemist. But of course, we do not know yet what the mechanism is. So only if we have compounds that are a bit more potent, we can then select for resistance and see where the resistant marker is, and have an idea of the mechanism of action. I see a question on pre-existing immunity to yellow fever for the vaccine from Dan. So yeah, it's not in the paper, and I didn't discuss it then, but we have beautiful data in mice where we vaccinate mice with yellow fever vaccine, and then 3 weeks later vaccinate them with the yellow fever SARS-CoV-2 vaccine, and we basically see, much to our surprise, I must say, that we see nearly no impact of the pre-existing immunity on the levels of neutralizing antibodies and on CMI levels. So that makes us quite happy.
No, that's really nice. Yeah, that's obviously been a limitation of the adenovirus,
yep,
some of the adenovirus vectors.
Yep,
that's why
definitely. I'm not saying that you would be able to go for 2 or 3 or 4 subsequent vaccinations, but at least in mice, and we'll do it as well in hamsters, we were, I mean, it's black and white. It's spectacular.
Yeah, well, that's very nice. Look forward to reading the paper in depth. So.
Thank you.
So what's your opinion on the predictive value of in vitro and animal models for COVID-19 patient efficiency?
Well, I think having an animal model is better than having not an animal model. And as I said, I mean, if we would have been able to test in March or February hydroxychloroquine, we could have advised against using it. Of course, I mean, none of the animal models completely recapitulates the pathology and pathogenesis. But for directly acting compounds, at least, I'm not sure if it should always, if the disease should always be exactly recapitulated in the animal model. I think somehow you can also look at a mice and hamster as sort of a black box on 4 legs, that on which you can basically have the virus replicating and just test drugs. But of course, I mean, I think the hamster model is pretty good. And there's a question also on the mouse models. The mouse models now, the ACE2 model or models, I think are pretty promising, and it will be. And we also are starting up these mouse models. It's more easy because you can put 5-6 mice in a box and only 2 hamsters. So we should be able, and others should be able to increase the throughput of testing drugs in animals with these mouse models.
I see that Christian has a question on the chat.
Yeah, the neurons in the hamster model, and we don't see an effect on behavior. We did not, I mean, we need to look into long-lasting effects of the infection in hamsters. So far, we didn't do that. We have been focusing mostly on the antiviral and vaccine studies, but this is definitely something that we want to look at. What I can say is that, and that's we have a paper in the revision and review in high-level journal. We do see also an impact on the heart of in hamsters infected with COVID.
So I think it's time to go over to the panel discussion now, and the questions left could be asked there. And please continue giving chat questions, which we will bring in the panel discussion. But now I give the word to Olli and the other three speakers.
All right, thanks Johan for moderating, and thanks everybody for wonderful talks. We have a really active chat here, and let's keep that rolling, and sort of by all means, let's continue also with the specific questions in that sense. While we wait for the questions to accumulate, I mean, let me first of all congratulate Johan on your Nature paper. That's really fantastic. And sort of I was just kind of pondering that how much is the impact caused by the oral route of delivery or the respiratory route of delivery versus other aspects of your vaccination construct?
I'm not sure. So what you mean is oral infection or the?
I thought that this would be administered eventually as a
No, it's like the yellow fever vaccine itself. So this is just sub-Q or even intramuscular injections vaccine. Yeah.
Okay, maybe I understood that wrong.
But you make a good point there, because well, there are quite a bit of teams looking at intranasal vaccination to rapidly get high titers of IgA in the nose. Now you can wonder with this high efficacies of the Pfizer and Moderna vaccine if this is still so much of a concern. I don't think that people, if you have 95% protection against infection, that this will be so much of a concern. But still, I think it's an interesting way of thinking.
Well, by all means, any one of you, please also ask each others, and sort of I have one point back to Daniel on the sort of a issue on the efficacy of the antivirals, and this relates to the point that Christian just raised on the neuronal effect and the long COVID. So is there any studies underway or planned that you follow these patients as to whether these antivirals help with the long COVID symptoms of patients?
Yeah, that's a great question. You know, obviously the biggest challenge we have in terms of long-term effects of COVID is the fact that all we have is 10 months of perspective on the epidemic to begin with. And so we're still learning what it means to be a long-term survivor of COVID-19. And I think whether, I would assume that antiviral interventions that prevent more severe disease would have a beneficial effect on some of the other manifestations of COVID, but that has to be proven, and there are studies planned to determine outcomes and correlate that with treatment. That is another place where conceivably some of the anti-inflammatory interventions may have effects that are not picked up on during acute hospitalization or acute illness, but have some longer term effect. I mean, just yesterday I was seeing a woman who had COVID in March, was never ill enough to be hospitalized, but still complains of fatigue and dyspnea. She was COVID negative by PCR, but still has a D dimer in the of about 1,000, and so that there's something not quite back to normal in her situation, and there are probably many others like that.
So, but as far as you know, there isn't a your internet connection is unstable. It's just, as far as you know, there's not a sort of a systematic long-term antiviral strategy to look at the long COVID syndromes yet. I guess it's too acute, too sort of the attention is elsewhere right now.
Right. No, there isn't yet an organized effort that I'm aware of. No.
All right. So let me kind of take a bit of a more generic approach here, and I think some of you alluded to this already as to what is the role of the antivirals and all of drug repositioning or drug discovery strategies in the future, when RNA-based vaccines are developed in a matter of a year, I mean, let's just go through all of you as speakers as to how do you see the lessons in that sense. I mean, obviously we don't know how it's going to play out in real life next year, but this has fundamentally potentially changed the relative balance between what's the time scale, and then what's the amount of efforts and money that we should put into the two categories. So you want to start, Dan?
Yeah, sure. I'll take a stab at it. Many years ago, one of the investment advisors for Yale University said that the best strategy involves diversification, timing, and opportunity. And I think that's true for a lot of scientific research as well. I think we need a balanced portfolio here, and clearly vaccination is the way to bring the pandemic to an end. But we don't know how long vaccine is going to be effective for, what the challenges may be of getting people revaccinated, whether we see immune escape over time and face an influenza-like situation. And there are still going to be people, whether because they're vaccine deniers or because they're less responsive to the vaccine, who will be at risk. And so I think it's going to be important to continue to have the development of additional antiviral therapeutics to be able to deal with vaccine failures or coronavirus resurgence, as well as to be prepared for the next pandemic, whether be it coronavirus or some related virus.
Mm-hmm. Peter, you care?
I agree on what's said, and I think it's you need to have diversity for many reasons. One is, okay, when we start the vaccine development, it's not always successful. I think that's a given in a way. But then also considering, as in our case, the access to the vaccines are kind of not equally distributed over the world, and there are challenges in distribution, etc. So I think there is a kind of room for kind of antivirals, and also considering we need drugs that can fill the kind of gap in time from the kind of rise of the disease and when you have the vaccines or other treatments available. So the kind of access to broad spectrum antivirals, I think we need to have that, and it needs to be ready. Clinically ready in the storage, basically. So you ideally should have a panel of different antivirals that you can kind of easily test when you have a new pandemic on rising somewhere in the world. I think that's the key.
Okay, Johan, anything to add?
I mean, Dan and Peter, you're pretty complete. So this is also the way I see it. Absolutely. I think this is a pandemic situation, of course, and the vaccines will, and I think we can be really happy that things went so fast and that the efficacy is so high. Still, it's a pandemic situation. There will be cases where antivirals will be needed. It's on the other hand, I was somehow afraid, let's say, beginning of 2020, that vaccine development would take much longer, and that we would have to rely on this repurposing drugs, because I said right from the beginning, and I said that all the time, repurposing drugs are not solving the problem. You can, and as Dan and Peter also alluded to, you can just perhaps alleviate a little bit with such drugs, do something with it, but well, just to put it into the context, let's look at favipiravir. If we look at the IC50 and cell culture, this is about 100 micromolar. If we look at the IC50 of the best HIV drugs, the best hepatitis C drugs, this ultra potent dengue drug that we developed, this is in a nanomolar and a picomolar range. So this is about 1 million-fold more potent than what you see with favipiravir. And people sometimes think that these repurposing drugs, that these are that these are real drugs now. I mean, it's something that you can just use for a little while to bridge, but you really need this, and you can do it. You can, against every single virus and viral family, you can develop ultra-potent drugs. You just need to do it. I mean, you need to invest time and money to do it. And well, I'm happy. I mean, so, you know, my lab is doing both things, antivirals and vaccines. So that part of my brain that works on antivirals is relieved that vaccines are coming, and that we do not have to try to fight the pandemic with this weakly acting drugs like favipiravir and others. But for the next, if I just think about my students, some medical students, the age of 22, 23, I tell them all the time, by the time you will retire, you will for sure have seen a pandemic. And I told that in December, and just a couple of weeks later, the situation was such that I was right. But I tell that, of course, every year. But I mean, we will, so by the time that my kids, which are in their 20s, will be retired, and my grandkids, hopefully, who knows, I mean, we will have seen pandemics, and we better be prepared for that. And we will need, as we discussed, as Peter, as Dan, as you, Ollie, also mentioned, I think we need just that weapon, these antiviral drugs, and it will need an investment. But if you put that into the context of the losses in life and all the medical problems and the impact on the economy, I think it's just a couple of $100 million is just peanuts if you put that into that context. Amen.
I think Johan raises a really important point with what you said about favipiravir, because that was also similarly true with lopinavir. Is
yep.
You know, you really need rational repurposing. Just because you can demonstrate some activity in vitro, you have to think about it in a pharmacologic context and,
yes.
and realize what's the achievable dose. It never made any sense to me that there was a huge opportunity cost in a way for all the trials, the people enrolled and the money spent on drugs that were very unlikely to really have an effect.
Yes.
And again, there was the imperative to do something in the pandemic setting, but it needs to be done intelligently. And I'm
yep.
a lot of
agree.
wasted time.
I fully agree.
Yeah.
Fully agree on that. And also sometimes, as I said, the imperfections of some of the repurposing candidates are easily forgotten. If you take favipiravir as an example, in childbearing females, it can't be given without contraception. So it's that's unthinkable at least in the parts of the world that we are working. You can't. This is impossible to.
At least in the elderly facilities where we will test it.
There it's safe, definitely.
Yeah. Well, they obviously, the repurposed drugs having to be used in combinations will also have combinatorial toxicity. That is quite unpredictable, perhaps, and not easy in that sense. But let me sort of push you a bit further on this route, on this two-path, the vaccine development and the pandemic preparedness, if I call it that way, via a collection of antivirals that are kind of a pre-made for the future pandemics. So if you consider now, well, I'm taking the remdesivir example that Dan showed, how enormously difficult, well, it hasn't been a COVID-19 drug to begin with, so it may not be the perfect example in that sense, but how difficult has it been to prove the clinical utility of it? Okay. So taking your sort of a point, Johan, which I kind of agree, that it would be nice to have this repertoire of pandemically prepared drugs ready to go and ready to use, but we don't know which one of the viruses will be the next one, and then having really a proof of concept, or not just proof of concept, but a clinical applicability.
I give
something like that. How do you make this happen in real life?
I give you an example. So we didn't publish it yet, but we developed a also ultra-potent enterovirus drug. It's active against any enterovirus against which we tested, including rhinoviruses, Rhino A, Rhino B, Rhino C, EV68, EV71, just name it, Echo, Coxsackie A, Coxsackie B. all in the picomolar to nanomolar potency. In animal models of antivirus infections, it's I mean, skip mice that you kill with just one PFU of Coxsackie, we can just treat them, sterilize them against the infection. So, and that drug it's a non-nucleoside analog. It's targeting the 2C helicase, and it just shows to me that with sufficient efforts, because this is not just something that you pick up in a screen and then test it in mice. We picked it up in the screen, and we with the chemist made 2,000 analogs before we had a clinical candidate. But it's doable. You just need to invest sufficient energy and money. And now, if a picorna, a new EV71 that is causing encephalitis, EV68 would pop up, this it's also in the polio endgame. It may play a role. This is the type of drug that you need, and resistance is not coming. We tested in skip mice, where we give treatment for a couple of days, and then we let, with the suboptimal dose, we let the virus coming back, we treat them again, and we continued for 2 to 3 months. We don't have resistance. And the reason is that we need at least 4 mutations to get resistance, and it just doesn't happen. So my point is, it's doable. I believe we have also examples against flaviviruses with non-nucleoside analogs, not yet published. There's much work to be done. I think for paramyxos it should be possible. And perhaps I'm too optimistic, but I think if you see the investments being made now in developing a vaccine, with a fraction of that, I think we can do a fairly big job against quite a couple of viral families in terms of developing antivirals. But
I think that's a great point, and this question just coming up in the chat about who should invest in it. I mean, I think governments should be investing in this.
Yeah.
If we had a series of compounds that had good in vitro data or animal model data, and we understood their PK and safety in humans, and they were there on the shelf to be used should an epidemic occur, you could then rapidly roll them out into phase 2 trials that would advance things very quickly.
Absolutely.
There were things that did get done right. You know, the recovery trial in England, leveraging the National Health Service to be able to study multiple interventions in different arms at the same time was a huge success, and it points the way to be able to do adaptive trials and avoids the problem of the epidemic dissolving before you have the chance to test things, which has always been the concern. When is the right moment to step in? You know, SARS-1 disappeared before there was the opportunity. The Four Corners virus from a couple of decades ago, similarly, the hemorrhagic virus. So, yeah.
So what are the diseases we should be prepared for? The pathogens, thinking of viral diseases, but maybe also bacterial infections or parasites.
Well, flaviviruses for sure, right? Look at chikungunya and Zika and blanking at the other one.
West Nile.
Yeah, well, West Nile, of course, yeah. But
there's, for example, in Africa, there's Wisselbrand, which is a sheep virus. It can apparently go over to man, and it causes also microcephaly in lamb. Just to name one, that's a flavivirus.
Dengue was the one I was trying to think of.
And dengue, of course, yeah.
All right. Well, I think that's a really interesting sort of a ending to our conversation. But before I end, I just wanted to see if you guys, the 3 speakers, would have sort of a advice to us, considering our drug discovery programs and so forth. We also have considered this pandemic preparedness concept as to whether that should include the drug discovery and drug development, drug repositioning angle, or whether we should just consider pandemic preparedness to screen for viruses and then expect that the global community will have these efforts and the vaccination programs will come up. But any thoughts for a small country with relatively sort of a focused efforts that are only making sense, because Sweden alone can't cure the whole world, but I think there will need to be an international consortium for sure developing these types of efforts. But any advice that you would give us in terms of, you saw a little bit as to what some projects are underway, as to what should we do in phase 2 of our COVID COVID-19 program when it comes to drug discovery aspects and drug repositioning and so forth. We didn't have time, obviously, to fully describe to you what's cooking. It's maybe difficult, but any sort of a immediate thoughts if they come to your mind?
I can say something, since we started up as many started, and so what became a limitation early on was access to infrastructure and you know, BSL-3 lab facilities that actually can do kind of in vivo studies, rapidly evaluate what are the kind of optimal kind of species. Is it mouse, hamster, whatever? What should you actually test your compounds in to understand, to build a kind of translational chain, so you and rapidly be able to kind of give people access to the screening cascades, to the test cascade that you need to evaluate compounds. You need to evaluate kind of different treatment options, etc. So I would think that at least for us took some time, because everyone was kind of desperate for these models, this test system. And also what we're seeing across is that there's not the kind of best practice. You see the huge variation in data published. Why is this? Is there enough understanding to say, okay, how do we produce reliable data? And I think that would have helped a lot in the beginning, before everyone was up to speed.
Well, any thoughts from the others, Johan and Dan?
You know, I think Peter said it right. Probably what surprised me the most was just how very simple fundamental things became so challenging. Nobody would have anticipated that we would have shortages of plasticware, shortages of gowns, shortages of guanidinium or RNA extraction buffers, because the demand became so vast and on such a global scale. And we really need a much more robust biomedical research infrastructure to be able to support this kind of work. And we really are dependent on it.
Yeah, just what I wanted to add. So you have seen the little video on our screenings platform. I had to fight for years with people to even believe that we need that one audit. I mean, not just for pandemic preparedness, but also for these many viruses for which we don't have anything right now. And so yeah, building the infrastructure too low, but Sweden that is, I think, in good shape. You have excellent labs, but just building the capacity to do this. If I always compare this, look at this nuclear bombs in which they invest, and the fighter planes. I think F-35 fighter plane. What is it? 100 million $100 million. I'll just put that into that context. I mean, the cost of a good SciLifeLab is just peanuts if you compare that to that.
That is an excellent comparison. And what I was wondering, and people were clearly impressed by your video about the facility, because we've been just sort of configuring an old BSL-3 with a few high content screening instruments and so forth. So it isn't quite in that scale. But did you get that done in just recently and as a result of the pandemic, or was this didn't exist before?
That's a story of 10 years, and we built that from scratch. It's completely this is unique. Nobody has it, and we got funding for that, obviously. And we built it, and 3 years ago, when we moved into this new building, it was 2.5 years ago, we were basically, it was installed, and then we needed 1 year of training the system. And let's say just a year ago, we were ready, and we have been screening on before 2020 on rabies and other bugs. But well, as soon as we got the virus in-house, we started screening on corona. I think this shows why we build it. just I always call it be prepared for the expected unexpected. And you can also turn it, be prepared for the unexpected expected. And I think these are just small investments for how to help protect yourself against these unexpected events.
All right, wonderful. With those words, I mean, I leave Johan with your words, prepare for the unexpected or for the unexpected expected. We will end this now. Time is flying. So I will obviously now thank very much for our speakers today. There was great talks, good discussion. Thanks for all the organizers. Thanks for everybody hanging out. There are 60 people still online now. So thanks for the whole event, and hopefully see you in another context. Thanks so much.
Stay
Thank you.
safe.
It's a pleasure.
Thank you.
Bye-bye.